All right, I think we're ready to get started. So good morning or good afternoon for those that are tuning in, and welcome to today's SSI webinar, Optimizing Preclinical Programs and Streamlining Clinical Translation. We're really excited for this webinar today. We hope it's informative to those participating, and it's really going to be focusing on such a critical phase in drug development, and that's the transition from non-clinical to clinical trials. You know, from bench to bedside, as they say so often. My name is Adrianna Valenciano, and I'm a Senior Vice President at SSI, and I'll be guiding us through this discussion today. I'm very pleased to introduce some expert panelists who will be offering their expertise on the subject, and they're bringing it from years of real-world experience. So I think it's going to be just very additive to those listening on how to practically think about this really critical phase. So I want to thank both Steve and Benit for joining today and welcome you guys to the webinar. Benit, this is your second time around. Steve, you're first. So maybe we start, Steve, with just a quick intro, and then Benit, maybe reintroduce yourself as well, and then we can jump into the discussion.
Sure. It's thank you for having me here. It's a little nerve-wracking being a rookie, but I think I'll make it.
You're well equipped.
Steven Zelenkofske. I'm a cardiologist by training. I've been in industry over 25 years, started in large pharma, have been a serial CMO 5 times now of startups of various sizes, from very small to medium-sized companies, and have done a number of filings across a number of modalities. I have worked very closely with both founders, CSO founders, as well as CDOs and CSOs to bring products forward. So I'm looking forward to this. This will be a lot of fun.
Awesome. Thanks for joining, Steve. Really appreciate it. Benit, would you mind reminding the listeners who you are and what brings you to this webinar?
Sure. So Benit Maru, Senior Vice President on the medical side with SSI, and I am an adult neurologist training background, and similar to Steve, although not as tenured, I guess, supporting early stage biotechs primarily in their non-development, human trial phases, and have been a CMO of a few gene therapy companies as well. So what brings me here, I guess, to round this out as well, is what we've seen actually more recently as well, and what some of the changes and challenges have been just even in the past 2-3 years, right, Steve, in terms of, the level of evidence that's required, not only from a first-in-human trial perspective, but also what's potentially sought after by investors, which are crucial to any development for any product.
Absolutely. Thanks, Benit. And for those listening in, we're going to have a little bit of discussion for about 25 to 30 minutes, and then we'll open it up for Q&A, which you can send in the chat, and we'll be queuing those up kind of at the end of this discussion. But really pleased to have both of you on the line, and for the reason that I said, just the real practical and real world experience that you both have had in this very critical phase of transitioning from a research organization to a clinical stage. And there's several organizational factors, there's the scientific challenges, and I think it's important for us to start the discussion with, what is the problem statement that we're going to be talking about today? So what are the common challenges faced when making that transition from research, your non-clinical to clinical trials, and what are some of the things that you guys have seen and are trying to solve for when you're in that point of development where you're saying we have great non-clinical data, we have great toxicology information that's going to inform how we progress this program, but what are the challenges that you think about as you're taking that next step and planning for integrated development? And I'll pose that first to Steve, and then would love Benit to chime in as well.
Well, it's funny. We've always, the golden ticket is really getting data in humans, and it's always been that way. But I think there's never been a time where there's been more pressure in being able to predict what's going to happen in human beings, and to be able to get to data as quickly and efficiently as possible as you can, as we are right now. We're in an unbelievably difficult time for biotechs, but even large companies. The goal is to really kind of decrease the failure rate and make sure that we can predict through. So that's really been the biggest challenge all the time. And it's about how do you identify early models and get involved early with these programs to understand what read-throughs you have to be able to understand how you build a program for the greatest success in the clinic, right? And that gets down to a number of things. We've always had challenges with what are the markers, what are the ways that we identify efficacy. A lot of these companies, and Benit's been in gene therapy, I've been in rare disease for the last several years, and before that, cardiology. And the hard thing is, we rely a lot of times on small animal models, and mice don't predict humans very well. They're great for identifying if you're hitting the target. They're great for identifying if you're hitting specific mechanisms, but the ability to read through the human beings hasn't been all that successful in a number of areas. And then you get down to the challenges which really make the program, which is who are you going to treat? How are you going to treat them? What are you going to measure? What's the dose? And dose becomes unbelievably important. And we didn't even get into safety, right? So I do think that the key to this is really kind of thinking about what the end looks like and understanding what you want to accomplish when you treat human beings, and then understanding your patient population, your disease manifestations, and your pathobiology unbelievably well, and how that translates, and having that crosstalk across the early animal work, the medium range animal work, through the research program, to be able to understand those things that you need to do to get there.
Mm-hmm.
Yeah.
Benit, anything to add there?
I think Steve's kind of covered it extremely well. I think everything that's been laid out are the things that we have to be thoughtful of and mindful of. And the last thing Steve said is essentially the end in mind. Where do you want to end up? Work backwards from there. I think the only one thing that I would add to that is, if this is moving from an academic venture into then a pharmaceutical organization, that narrative, how you position that narrative, just be mindful of that, because different groups will want the same narrative, but how you position it to them is going to vary ever so slightly. An investor will want a certain pathway to that, right, Steve? Because he's smiling. So I think that would be the only. I think that's actually one of the other challenges, aside from the non-clinical to clinical, is where do these academic founders, how do they translate what they're doing, to an investor? So that would be the only one add, I guess.
Yeah.
Yeah.
And I was going to add to that you got to remember, you always need great experiments, but great experiments don't always make great drugs. Right. And so that's the thing. And we're all scientists at heart, whether we're founders, whether we're academics, whether we're, chief scientific officers or chief medical officers. And in a lot of ways, we romanticize about the science, and sometimes you have to take your heart out of it and put your brain into it and sort of plan a little bit. And so that's the other piece that's hard.
Yeah.
Sorry to interrupt, Adrianna.
Oh, no problem. I was going to kind of maybe take one step further into some of the discussion you guys mentioned about kind of the animal models and kind of the translatability there. And obviously there's been so much research that, as you said, Steve and Benit, that doesn't recapitulate to in humans, even though there's fantastic animal data. And maybe you guys could offer some guidance to some of the folks who are listening about how animal data and some of that non-clinical work should be considered as you're progressing into sort of a development approach and a development plan, because I do think that it can be a crutch, and there can be some things that people are, said, well, we saw this in the animals and we will see it in humans. And that's just maybe not an assumption that we're able to support in all cases. So I'd love to talk to you a little bit more about, really how can you use that animal data, and how do you apply it for development planning? Maybe Benit, you could start with that.
Give it.
Sure. So I think there's several layers to that question, I guess, Adriana. I think the first thing, going back to what Steve said, was understand what your population is. What is that final group that you want to treat? Then work backwards from there. Now, there may be several animal models, there may be one animal model, there may be none. So I think understanding which animal model is most appropriate, if there is one, for the disease that you're targeting is actually extremely important. It may not recapitulate 100%, and taking the example of gene therapy, you have a knockout model, you actually have other genes then, which upregulate or downregulate within the system, of the animal, for it to survive. That is a confounding factor you need to think about as well. So try to think about what hallmark feature of the disease that is being recapitulated also. That's one thing to think about, whether it be, if it's, optical, is it something to do with a retinal layer? If it's to do with, something neurodegenerative, you need to think about the gates of the mouse, for example, whatever it may be. But I think if there is, try to whittle of the plethora of things that you could choose from, try to narrow it down to one or two things that you can really hang your hat on, and how that then translates, from what your first in human population may be, or not even first in human if it's a safety study, but what your first, treated population would be. I think that would be the first thing to think about.
Mm-hmm.
Yeah, and I was going to say that, it does, what Benit talks about gives an argument for both natural history data, understanding populations, but biobanking and a number of other things that you want to get from patients. But, I want to talk about the other side of this, which is sometimes we confuse target engagement with reads through for efficacy. And we have a lot of examples where the biomarkers in the animals are very, very easy to translate to humans. I mean, when I worked on the hemophilia B programs, factor levels translate very nicely, and you can actually use them to predict very, very cleanly how you're going to do in human beings. And I think those are no-brainers, right? Mm-hmm. But there are a lot of areas I've worked in that have been a lot harder. These progressive, long-acting neurodegenerative diseases are tough. Even in cardiovascular, it's been a little bit hard. And in kidney, it's been a little bit harder. And so we spent a lot of time and money trying to delineate in human beings what are surrogates and what are biomarkers that read through early in humans, and then how you can how you generate, recapitulate disease models that allow you to use markers to be able to do that in your early animal studies is unbelievably important. But sometimes we go, when we're trying to hit we believe this is a target that if you engage, you'll get a benefit. And then the measurements you make in the early animals are really measurements of target engagement and not necessarily of predicting efficacy. And I think you need both, but right. You have to remember that just because you get target engagement doesn't mean that you're going to have efficacy. And understanding what the pathobiology is in people.
Right. And
it really does speak to doing some extensive, getting early into your population and understanding that if it hasn't been delineated.
Right. And that's a really great point, Steve, especially obviously with particularly the rare disease area that you've been involved in. Obviously tons of patient heterogeneity, and the pathophysiology can be observed differently. But I am curious on how you utilize natural history to help inform you in terms of your development strategy. How have you been able to kind of marry that with your non-clinical data to kind of tell a more complete picture, and how have you been successful in doing that? And that question could be for both Benit and Steve, because I know I've worked with Benit on a lot of natural history projects, and how important that data is to help inform both your non-clinical but also clinical strategy. So maybe we start with Benit and give Steve a moment to pause, and then we'll get back to you, Steve. Since
Thank you.
The question. Yeah, absolutely.
So I do think, understanding that patient population, and again, I apologize because I'm going to be speaking more about, an ATM beam gene therapy in this scenario as well. But, rare diseases, ultra rare conditions, there is, a heterogeneous phenotype. Depending on the type of mutation said patient may have, they're all going to end up eventually with the disease, with the pathophysiology. It's just at what time interval do they get there? That's something that, does vary from patient to patient. So you will get the question from the regulators, like, how is that patient appropriate to treat? Is that missense mutation actually a true disease? How do you know if that disease, if that mutation is disease causing? That's where, biobank information comes in, all the genetic data that exists, literature comes in to support you. But going back to the question on natural history, understanding what data you have, so understanding what the patient profile is, there may be some scenarios where patients will exhibit a certain phenotype. At what time point do they exhibit that certain phenotype? So it is, in a way, trying to find something homogeneous in a heterogeneous population. And that's what you can definitely leverage natural history for, as one example. Another thing, if we think about ocular diseases, there's retinal thinning. If there is the data available, you can start to, see what data has been collected, whether it be through, OCTs or ERGs and things like that. And then actually, if you can start to see when the retinal layer starts to thin and what the rate of progression of that disease is, that's actually really helpful to know. There's other scenarios where somebody may have a seizure, a developmental epileptic encephalopathy. It's a seizure disorder, but it also then comes in with other neurodevelopmental components. There's, delays of learning, loss of words. People try to correlate seizures in that example, to cognition. More often than not, if you actually look at the data, that's not true. The cognitive aspect is actually one separate, distinct part of the disease as well. If you know where there are gaps, you could try and help fill the gaps on the natural history. That's one point. But the other point is just understanding what those nuances are. And then going back to your question about how do you use it, is when you start to find these trends, you then start to see if you can then fill them in an animal model, using the ocular example. If it is a certain retinal disorder, it's fairly certain that actually if you looked at the animal model, whatever it may be, whether it be a mouse or a canine, you may see the retinal layer thinning as well. And that starts to then show you what the correlation or similarity, or translatability, I should say, between a human and a mouse or a dog is.
Right.
Some of these seizure disorders, they do have mouse models, but they only recapitulate the seizure aspect. They don't actually recapitulate anything in the developmental aspect. So it's understanding what those differences and nuances are as well.
Yeah, no, definitely. And I feel like what we're like kind of cornering in on is this discussion on translatability, and really how does it translate and or doesn't it, right? And I think, I'd like to maybe Steve, you can weigh in on this. What are some of those key areas that need to be considered in terms of translatability? We talk a lot about endpoint selection. There's the animal age. There's the route of administration. So, as you're thinking about your CDP and you have this very valuable data in the animals, how do you practically translate that? And maybe we could start with endpoints and how you think about it. Obviously, the natural history and the way Benit described using it is definitely a factor and will help you identify like what are the things that we need to that we observe in the human that we want to make sure we're addressing as a part of the clinical trial. But I do feel like there's it's a very technical question, but also probably a strategic question. And how do you translate?
Yeah, and actually, I'm going to give you a few examples because I think it's all different for different programs, right? So, it's funny. I used to say I used to watch that movie City Slickers, and he would say to I always give examples like that. So, used to Billy Crystal says, to Jack Palance, do you know the meaning of life? He says, the meaning of life is one thing. The whole movie is trying to find out what that one thing is. At the end of the movie, he says to him, the one thing is different for everybody. Everybody's got one thing, but it's all different, right? Yep. And so I look at that as sort of these diseases. It's not one size fits all. There's one thing in every disease. You just got to figure out what that one thing is.
Right.
So it's funny, doing a rare cardiovascular program, we really did a strong natural history database with an academic who was collecting a lot of data and collected a lot of biomarkers and imaging markers and understood the longitudinal development and how the disease manifested. And then when they went to do the animal model, the mice developed hypertrophy early, but then developed dilated hearts and died, relatively quickly, and didn't really follow the course of the disease. And we had to go back and look at different knock-ins and knockouts to be able to recapitulate the disease. And if we had gone with the model that we had looked at the beginning, we probably would not recapitulate or test a drug in a disease that, in an animal model that really told us anything about what was happening in the disease. So we used the natural history data to go back and look at the animal model to see if it was really manifesting the course of disease that we wanted to test. But the other was that we actually used 3D organoid modeling as well, and used a live 3D organoid to be able to fill in those gaps that we knew we weren't going to get from the mouse model. And we think that will predict well into human efficacy. So we really triangulated a program by understanding the disease nicely. I've worked in a progressive neurologic or neuromuscular disease where the disease manifests itself by loss of conduction in the adults. And again, looking at the natural history data, thinking early on that we could use electrophysiologic data as an endpoint in patients. And what we found is that the patients lose their sensory electrophysiologic function really, really early, and they either have it or they don't. It's an all or none phenomenon. The ability to be able to use that as an endpoint becomes difficult, but motor conduction does change slower. So we went back into the animal data, and we're now looking at motor electrophysiologic and motor conduction. And so the ability to be able to understand how this manifests and what you're going to use as an endpoint in patients and how it manifests in patients, and it translates to long-term outcome. And then whether you can make that measurement in a model. And it was real interesting because in order to do conduction, the mouse models, you can do it, but really what we were trying to do was do it in somewhat of a larger small animal. So we tried to do rat, but we couldn't get the same behavior in the rat that we did in the mouse of the disease, and ended up having to go back to the mouse model. So it's a couple of examples in real life of how you look at an endpoint, how it manifests itself in people, and then how you go back and reevaluate the models you're using to be able to translate better. So I think that in and of itself allows you to be able to feel more comfortable that you're doing the right thing. Route of administration you raised is really, really important. I've done programs where we've done IV administration in gene therapy, where we've done direct administration into the brain in Huntington's disease in a program, and then intrathecal delivery in some of these progressive neurologic diseases. And being able to understand how that how that's done in a human, what kind of the best ability to administer, and how that behavior will occur. And then when you do your animal work, being able to do something that shows you how that's going to behave when you translate over to people. So, when we did intrathecal in the mouse, we were getting tons and tons of systemic exposure, and you get a lot of backflow and a lot of the animals administering intrathecal in a mouse is not easy, and it's technically hard, and reproducing that is unbelievably hard.
Right.
You try to do what you do in primates, but running into problems with primates again. If you want to give a bolus administration to human in CSF, you're doing it into 120 or 130 mLs, but when you do it in a mouse, you're doing it into 12 mLs. So you need to really be careful about the time and how you administer it and what kind of adverse events you get. And so the ability to be able to understand, again, in humans, how you want that administration to occur, and then to be able to find the right animal model to be able to predict how that translates to things in gene therapy, let's say biodistribution, and ASOs, it's biodistribution, it's transduction in gene therapy, all of those things. So, it gets back to being able to use something that gives you a sense that you have translatability in that animal model, but you really need to know how it behaves in the human.
Absolutely. And Benit, I'd love to kind of raise the same question to you, but maybe add an additional lens for some of the regulatory folks that may be listening as well. And how do regulators think about translatability, and maybe some examples where you've had to think really critically about it and present it to different agencies, and what are some of the areas that they're very focused on in that translatability paradigm?
So I think, I think this is one important note as well. Depending on the type of therapy it is, whether it's an ATMP or a small molecule, especially on the ATMP space, there are differences and there are nuances in what the FDA will require versus Health Canada versus EMA, MHRA, and others also. I do think that's just one important piece to remember, that different agencies will require different things. The one other component that does come up is if you are targeting a disease area, and you're saying that this manifests at a certain age, the age that you're treating the animal does come up fairly often as well. And sometimes somebody is going to dose P1, P2 in a mouse, so postnatal day 1 or 2. If you look at like the neurodevelopment and what the structure is and how the brain is formed, that's a child that's still in the womb, for example. So there are some of those nuances that do come up, so we just have to be slightly mindful of them. You can always then think about the argument that you're making in terms of if something is going to be present at birth, then the P1, P2 dosing doesn't necessarily make that much of a difference. But if the manifestation of the disease is several years later, you are going to struggle with that argument of dosing an animal that early. So just that's one thing I would say that is important. It does become a matter. There's a lot more nuance when you're looking, and I say this in air quotes, softer endpoints. If it's a matter of life or death, that's a much easier argument to make than it is if you're it's like a loss of function, right, Steve? If there's things that happen progressively over time, just thinking through how that would be. So that's just something very important to keep in mind. And if you are going to struggle because let's say the animal dies within a month, and it's just going to be difficult to treat, then the mechanistic argument has to come in as well. You may need some additional in vitro data to support the argument you're making as well. So it may not all just be driven by animal data. Think through what the gaps are in what you're presenting, and then you may need some additional in vitro data to support your non-clinical in vivo data as well. But again, it depends on the jurisdiction that you're targeting.
Right, exactly. And I think that just one last question before we open it up to the group. I'd love, both of you have been involved in growing organizations that have gone through this pivotal kind of transition. And I'd love to hear, maybe we could start with Steve, talk through some of the things that from an organizational perspective need to be considered as you start expanding, growing new functions, growing new capabilities. And what are some of the I would say lessons learned that you've had when kind of growing during that time? You're obviously cognizant of funding, you're trying to be austere, not overspending, but knowing that you need to make some very critical investments. So maybe a lesson learned or two for those who are listening in who might be approaching this or could use the guidance.
Sure. So one of the things, a lot of companies do is they think, well, we don't need medical people or clinical development people until we get to the clinic, right? Or close. And, I find that, a lot of times you come into a program that is ready for IND, but not really ready for the clinic. And in particular, you haven't really done all the stuff we've talked about. Now you're behind the 8 ball. You don't know the natural history. You don't know the endpoints. You don't know the population. And you've got target engagement, but you don't really have, anything, your biomarkers, your assays, all of that stuff developed. So pearl number one, I think, is you really do need to start thinking about medical input early in these programs. I think you've got founder CSOs, but when you go to hire CSOs, you should be thinking about, how do I supplement or have somebody that's either a CMO or a temporary CMO or rental CMO or somebody in there that really understands the disease that can be a active partner. And I think the other piece of that is that people underestimate the relationship that the clinical development folks have with the research folks. And when you build that relationship and that duality early, and you work to work through the initial push and pull, people, it's a different language, and it takes a little time to learn to work together. But I think when you've got a CSO and a CMO that work extremely well together, programs tend to be a lot more successful. I think the other piece of this is, and it's thinking through the operations of how you're going to pull some of this stuff through. And again, a lot of times what ends up happening is you need stuff that you have to have for the clinic that has to be developed early and has to be evolved over the time of the research program. Assays are a perfect example in rare disease and gene therapy, where the assays don't exist and you need them to either qualify patients or follow safety or something like that. And you need them at the time you start. And I've had companies that have waited to do this work, and then all of a sudden you now have an IND and you've got 9 months or a year of developing an assay and you can't dose your first patient. So thinking ahead, planning ahead, having the medical input, the clinical development input, and to think a little bit more about operational input is extremely important, so you can start really having a seamless move from filing your IND. One of the things I talk about is it's kind of like GPS, right? You don't put in your GPS, I'm going to the corner of the block. You say, I'm going to this place, and it tells you how to get there, make a right turn, make a left turn. And we tend to think, well, if we can get the IND or we can get to the pre-IND meeting, we're good. And then, I look at that as part of the way there. So we really ought to be thinking about what's the destination and what are the tools we need to be able to make those right and left turns to be able to get there.
Absolutely. Benit, any insights or thoughts you have about also scaling an organization, and what are some of those early investments? And, Steve hit on the kind of the medical input and that really clinical expertise, but any other areas that you've been seeing as a part of your clients where there's scale is required as you make this critical transition?
Yeah, I just one thing to add to what Steve, I agree with everything Steve said. And it's like, for me, fundamentally is, do not let perfection be the enemy of good. And I think that in this setting, where things are quite agile is actually quite important. So other than medical input and, the cross-functional work that's needed, I do think, to Steve's point, what's that final destination, that end in mind, really do need to think about, to me, it's patient access, not market access, right? How are you going to, yes, you may get your regulatory approval, but then you need to get, whether it be an HTA or payer piece, if it's an ultra rare disease, if it's a gene therapy, that type of work probably needs to start sooner rather than later, in my opinion, and in my experience. And that's probably the only add on top of the medical piece that I would think. I think cross-functional work is extremely important. And also, it's okay to say, this isn't my area, let me pull somebody else in to help me, because a CMO is required to be lots of different things. You can't be good at everything, right? And it's sometimes just acknowledging your limitations as well. But I do think that end in mind thinking, and if we're doing it for the patients, and that's what this is all truly about, it's going to satisfy the investors regardless anyway. So think about that, I guess, and I try and bring some of that in sooner rather than later.
Absolutely. Absolutely. Thank you, Benit. And thank you, Steve. I think we're ready for some questions for the audience. I'm just going to give it a second as they queue up. Right. So one question that we have coming in is, how can researchers proactively de-risk their asset and anticipate potential roadblocks in the transition journey? I like this question because it's, really focusing on the de-risking, and I feel like that's a common terminology used as a part of development strategies. What are we doing to de-risk? I know that from some of the experience I've had with clients, they're they're not just thinking about, they're obviously thinking about their non-clinical studies and their toxicology work, but they're thinking of other important trials that can augment and supplement their overall package, and that helps de-risk. So I'd be curious if you have other, maybe we'll start with Benit, any specific tactics or approaches that you would think would help to de-risk as you're making the transition from non-clinical to clinical. Any thoughts there?
So I think one of the biggest things that I would say is, do not be limited by which jurisdiction that you really want to start your your first studies in. Just, some jurisdictions do want more data than others. So that's the first piece. And actually, Steve made this comment earlier, the trade is in human data, with the greatest respect. The sooner you can get human data, the better it is, just not only going to be for the program, but for the company. And that's kind of the key. So just be open to going to different jurisdictions. Just because you started in one country doesn't mean that, you can't have, your first launch and reimbursements in another country. I think that's one of the biggest things that's really worth thinking about, because, we'll take editing as an example. Looking, editing, gene editing holds, quite a lot of hope. There is much more restriction and much more data requirements that are required around the FDA, where it does seem to be slightly more open, and things are slightly more amenable outside of the US as well. I mean, that's just one example, I guess, in terms of novel technology that's coming through. And where there may be some differences, I guess, from a jurisdiction perspective. So I think that would be the first thing that I would think about, just from a de-risking perspective.
Right. Absolutely. Anything to add, Steve?
Yeah, so I agree with you completely. I think the other thing about it is that whatever you want to call it, the integrated development plan, preclinical to clinical TPP, having early TPPs, having an early IDP, milestone-based driven development is extremely important. I mean, at one of the places we call them swim lanes, the other place we call them checkpoints, whatever you want to call them, right? But having the ability to be able to have go, no goes and make decisions and understand what your go, no goes are early and what you're trying to do. And again, it gets back to what Benit was talking about. What's the end game? And then how are you going to get there? But what are the milestones and the measurables and what your programmatic KPIs are, whatever you're calling them, are for each of these things, so that you understand what your decisions are. They don't have to be set in stone. They can be very fluid. I remember one program I was working on, we had 4 things we had to achieve to make a decision, and we found out that 2 of them were unachievable. So it gets hard, because it gets hard. But the 2 that we got were, very convincing. We moved on. And, sometimes when you have to make clinical decisions in your programs, you're looking early on, when you've got, especially diseases that take a long time to develop, you look for directionality, right? You look for a bunch of stuff all going in the same direction. But preclinically, you want to be able to understand what's your decision points. And I would not forget about drug, right? Drug becomes, CMC is unbelievably important. Most of the time you get messed up by drug, and not just messed up by drug early and not for your IND, but, I've done clinical programs where, drugs become a problem. Just supply chain is the headache, right? So, I mean, it's a parallel path. You've got efficacy. We didn't talk at all today about safety, and safety is unbelievably important, and we should probably talk a little bit about that. And then don't forget about drug.
Absolutely.
I agree. I mean, safety is paramount in all of this. Just to add one more thing, I guess, to the integrated approach, I do think trying to ensure standardization across what you're doing non-clinically to clinically is also something that's actually extremely important. So Steve, you talked about assays, right? If you're looking at an enzyme, you need to make sure that that assay is ready. It's good to go. It's good to go from the animal study setting right the way through, to the human setting as well. Looking at again, use the eye as an example. If you want, make sure you've got standardized OCTs right? If you can have the standard same device is actually quite important from the non-clinical setting, make sure everything's standardized. Then when you go to the human setting, try to be as judicious as you can with regard to which centers you choose. And this is where clinical operations actually becomes really important. I know that there's a question around clinical operations and early engagement. Only certain centers will use certain equipment because you need to make sure that there's standardization in what you're doing, again, from an OCT reader. And that actually needs early ops engagement in terms of, where you're running the trial, if the centers can do what you're doing, if the equipment's the same, you need to ship them out. So I think, trying to link that standardization piece as early as non-clinical to clinical is actually something that would help de-risk overall.
Absolutely. Yeah, and I feel like just something to add to that, I think we had a question about early clinical operations engagement. And, this is a really, Steve, I'd love to hear your thoughts on this. Oftentimes we see organizations engage clinical operations and start really building that function once they are really, creating protocols and synopses and starting to, actually crystallize what these trials are going to look like. But maybe you could give us some insights on, how some other proactive organizations are thinking about ClinOps much earlier, to the point Benit made. If you're using something, measuring something as a part of your non-clinical program, how is that going to, how are we going to advance that forward? And is it practical and operationally feasible? So any thoughts on that?
Yeah, it's interesting. I think getting ops involved early is important because I think you really need to think about how you're going to do stuff. And all the things I just talked about, which was, the drug supply, the ability to get assays through and all that stuff's important. But I think the more non-standard your program is, and the more you're doing stuff that isn't, giving a drug intrathecal or into the brain or into the organ versus IV, creates different challenges that need to be thought about early, and where you're going to do it and who you're going to do it with and how you're going to train people and how you're going to get the stuff that you need to do, whether the equipment has been approved or not, whether it's approved for that use or not becomes important because you got to not only have regulatory, but you got to have people on the ground if you're outside the US to start thinking about, well, we don't have a C mark and now we're in Europe and we got, it creates all kinds of complexities.
Right.
So you've got those issues. Traditional assays and safety labs are easy, but when you start thinking about non-traditional assays, non-traditional markers, how you're going to do them, how you're going to not only bench validate them, but get them into humans and be able to get it, transfer them over to a clinical vendor and be able to, how you collect that blood and what you're going to do, or how you collect that body fluid, not just blood, and how you're going to do it is unbelievably important. And, I've been doing this a long time and I would never dare to do it without a ClinOps professional. Yeah, it's do not operate this machinery by yourself, right?
Yeah, absolutely. So,
I've become so reliant on having somebody who's a ClinOps professional with me, being able to look at this and saying to me, okay, you say you're going to do this, but this is not just like checking a box and doing it. There's a lot of stuff we have to do here. So really thinking about there's translation and there's translation. So the translation of being able to say, what's the science and how does it translate to a person, right? And then you say, translating, what are you doing in your animals and how are you going to translate these to people and how are you going to pull it off becomes unbelievably important. And when you start to, you should be thinking about that early. And when you think about that early, as soon as that comes into your mind, you should have a ClinOps professional next to you telling you how you're doing it wrong.
Yeah. Well said, Steve. Well, that's definitely salient words to hold on to for sure, for those listening. I think we're coming up at the end of the webinar, and I just wanted to close out with maybe just final reflections from Benit and Steve. You both allude to really thinking with the end in mind. In our next webinar, we're going to be talking a little bit more about clinical trials and really some of the things that need to be considered as a part of developing, discrete studies, but also a part of this end in mind and kind of what is the ultimate goal. So I'd love to just, I'd love that you use the analogy, Steve, about, the final destination, and there's certainly different turns along the way. And one of them is, getting it right with your non-clinical work, as well as your IND enabling and other toxicology work. But maybe just some final reflections you have on, why this early work is so critical to helping you shape what is ultimately kind of a clinical program that will meet those needs of patients and really address the unmet need. So maybe Benit, you could start, and then we can have some final words with Steve as well.
Sure. Yeah, and look, personally, there's an unmet need for a multitude of diseases, and as a clinician, we want to help people. And for me, the driving factor is how can we appropriately and efficiently and effectively get this, to human as soon as possible. And to me, that's kind of always been the driving factor. And let's think, sometimes you kind of just need to think outside of the box a little bit as well. Again, perfection being the enemy of good can be a stumbling block if you come from academia, because, you want to do everything and everything and everything. And that it's good, but it can also then cause delays potentially as well. But I think, focus on safety, to Steve's point, ensure that you've got the right dose, make sure that, you've got an early TPP, because you want to know what your final destination is as well. I think that's kind of
excellent. Any final words, Steve?
Yeah, so we're all really smart people, excluding myself, but we're real people, and sometimes you can be too smart for your own good. And what I find is that the programs that are most successful are the ones that are elegant, well thought out, and as simple as possible. I mean, how many times have you looked at something and said, my God, I wish I thought of that. That is the simplest thing. Or you've taken something that's unbelievably complex, theoretical physics, and the smartest person in the world can make it sound like it's the simplest thing to explain it. And so, the simple thought about this is, how do you be able to think through all those components of your program, understand it, break it down to simple facts, and then put it back together with simplicity so that you can actually do something that's not so complex that you can't pull it off. And I think if you can do that, those are the programs that really do well. And it's funny, I worked for a development person who used to pull his team in and say, if you say to them, what's going to go wrong, they almost tell you nothing because they think they have everything covered. And he would say, imagine we're done the program and it failed. What are the most likely things it would have failed for? And then they go back, they do a pre-mortem, and they go back and they look at it and they start to plan around that. And I think it's a really good practice start thinking about how you're going to get there and what you do, but to not make things so complex that you can't pull it off. So,
Thank you.
Keep it simple.
I love those parting words. My takeaways at this very moment are simplicity, translatability, and bring your ClinOps person. So if I could sum those up for you guys. But no, I really appreciate this conversation, Benit and Steve. It's been really great, and I hope it offered some real practical guidance for those who are listening in. In our next webinar, we're going to be taking a step further and kind of talking through central themes of pivotal clinical trials, and really talking through what are some of those challenges, as well as best practices that we can discuss as a team. So again, thank you to Steve and Benit for all the great guidance, and to those who sent questions in. We really appreciate it. We will speak again in the new year, and hope everyone has a great holiday season, and we'll talk again in 2024. Take care, everyone.
Thank you, everyone.
Thank you.
Take care.
Bye.